All right, so our next speaker is going to talk about predicting the fine structure of cis-regulatory elements.
Okay, thank you. This work is basically the thesis project of a graduate student in my lab, Dongwon Lee, who's very talented, and a graduate student who just finished, Mahmood Gandhi, who's now at MIT and is a postdoc and developed the gapped Kmer extensions of our earlier work. And I'd like to mention that we are in the Department of Biomedical Engineering at Hopkins, and I'd like you to encourage your mathematical and computational grad students to apply to our program, where we're nurturing the next generation of computational biologists. So Len gave a great introduction to my talk. I just want to underscore the fact that the majority of the GWAS-associated SNPs with now several hundreds of traits are mostly found not in exons, but in intergenic regions, are not linked to exons. These are enriched in DNA functional regions, hypersensitive regions, and there are now more and more a steadily growing number of examples of variation in TF binding sites that are associated with these phenotypes. So our approach is to use large ENCODE data sets like the ones that Len described in mouse tissues and in human cell lines to train a computational model of the mammalian regulatory code. So what we mean here is that we want to know, what sequences within an enhancer make it function in which cell types at what time in development, and also what mutations would specifically alter the function of those elements. So this is a picture. I guess they're all a little bleached out. Kind of unfortunate. Can maybe I adjust? If all day long the pictures will be bleached off, maybe we can make an adjustment now to this computer. Does anyone want to give it a try?
Stand closer here because there's feedback if we move it.
Okay, no problem. Sorry. I see. I think I can. I guess it's the projector. Okay. Sorry about the bleaching of the photos. So this set of experiments showed that a wild type, while wild type HNF4 alpha binding was different in human than in mice, in the analogous locus in mouse, that a chimeric mouse carrying the human chromosome showed a very similar binding pattern. And this importantly tells us that the evolution, the differences between mouse enhancers and human enhancers is occurring at the level of the DNA sequence and not at the level of the transacting factors. So if we can understand how enhancers are functioning in mice, that same set of rules will to a large extent apply to humans. So this is the same picture, believe it or not, that Len showed. So our initial work on this problem, which was published in December of Genome Research, we predicted limb and forebrain and the midbrain enhancers identified by Len's group and reported in this paper just using DNA sequence. And so the way we did it is we took these DNA sequences and we don't know what transacting factors are active in these different tissues and what are causing these to be enhancers. So we just counted the frequency of all 6 MERS in these set of sequences, and we used these vectors of Kmer counts to try to build an SVM which could distinguish between the enhancers and the random genomic sequences. Okay, and the result of this classification is a list of Kmer weights that describes the black boundary that you can't see between these sequences, which I can see very well here. I apologize. There's a lot of data in my talk that you'll just have to take my word for. So using this model in 5-fold cross-validation, we can predict the forebrain and the mid and limb enhancers versus random genomic sequence. We can also predict the different sets of enhancers versus each other. And the way this model is doing it is by identifying large positive weights for binding sites that are active in the specific tissues, but also sequences that cannot be present in those enhancers. So since this paper in Genome Research 2011, we've applied this methodology to other data sets, and we can predict all the ENCODE P300 ChIP-seq data and DNase data in mouse and human cell lines with AUC, median AUC of 0.94. So we next wanted to dissect the enhancers and try to understand what regions within an enhancer were contributing to our ability to be able to predict the sequences. And the first pass through, as you can see in gray, thank you very much, right? We were kind of disappointed to see that the signal at each base along a locus that was DNase hypersensitive that we trained on, the gray signal using the Kmers SVM, even though it was able to predict the sequence overall accurately, the specific signals within the sequence were very noisy. So since then, we've developed a new model, which we call the gapped Kmers SVM, which is shown in red, which has a much clearer distinction between the positively scoring regions in the sequence and the kind of background levels scores in the sequence, and that these high scoring peaks align very well with DNase digital footprinting, and also with known transcription factor binding sites. So the whole region is DNase hypersensitive, but there are regions that are protected from DNase hypersensitivity by the actual factors that bind in this enhancer, and there may be 5 or 6 or 7 in this region, actually in this promoter. So the new development is to use gapped Kmers as features to predict the most likely estimate of the Kmer counts in the training sequences. So the idea is that the SVM works very well by predicting the boundary between these elements using a small set of support vectors. And that's great to avoid overfitting in the SVM, but it has a problem that when you calculate the weights from these support vectors, there are fewer examples, there are fewer training samples, and they're noisier, the sequence features. So instead of using the actual counts of say, Kmers in these sequences as features, we can look at the gapped Kmer counts, which are much more robustly estimated. And there's another set of estimated counts that produce the exact same gapped Kmer frequency distribution, which is flatter or more likely or higher entropy than the actual observed set of training sequences, of which we only have one copy. So this is kind of a better representation in some sense of the whole set of functional features. And what we see is that where the Kmer SVM, the Kmer-based SVM, is susceptible to overfitting with longer Kmers as the counts get lower and lower, the gapped Kmer SVM has high AUC for up to K of 20. And so it estimates the specific features within the enhancer with much greater accuracy. So we next wanted to directly compare the gapped Kmer SVM score with functional SNPs. So we looked at DSQTLs, which are regions of DNA signal that are correlated with genotype at a SNP, right? And we wanted to see what does this change from a C to a T that causes this drop in DNA sensitivity and disrupts this NF-kappa B binding site and ChIP-seq signal. How does this change reflect in our score? So the red is the gapped Kmer SVM score across this locus, and the single change from a C to a T changes this prediction from red to blue. And if we do this for the full set of DSQTLs, we get a much bigger change in the predicted SVM score than in random regions of the random SNPs. Further, we've shown that experiments validate that luciferase expression change is consistent with the predicted change in the SVM score. So this shows that the SVM scores that make a large negative change have much less luciferase expression. And we next wanted to look into, how is this working? So we looked at not a set of complicated enhancers, but just binding of a specific TF, MYC, in 5 different cell lines. And we found that we could predict these regions, as I'll show, and they basically fell into 5 classes: common MYC binding peaks, which are mostly in promoters, and then tissue-specific binding events. That's, for example, here's a binding event that's in HeLa and absent in all the other 4 cell lines, right? So when we tried to predict these regions with the classifier, whether we predict against random sequence or whether we try to predict one set versus all the others, we get high AUC. And the features, when predicting against the other regions, now aren't MYC binding sites, as are found here, but cofactors, such as AP1, which is active in HeLa, and HNF4 in HepG2, ETS, and GATA. So when we look at the DNA sensitivity in these regions, we get a good agreement between DNA signal, which was not used to train the predictor, and our prediction. So here are just a couple of examples of a common peak which we predict. This is the common signal versus background sequence. This is the common, these are the SVMs trained against all the others. And this is a common bound region in a promoter which has a binding site and promoter-specific sequence factors. This is a region which has a HepG2 active MYC binding site and is only DNase hypersensitive in HepG2. This region is predicted to be specific to K562 and is only DNase hypersensitive in that cell line. So the SVM now using gap Kmers has a lot of features. And to aid interpretability, Dongwan generated a set of PWMs from these high-scoring regions. And he showed that from the common peaks, he finds specific PWMs for MYC, ALK1, SP1, and kind of promoter-specific factors that are all conserved, shown in red, relative to flanking sequence, and also show reduced DNase hypersensitivity consistent with footprints. Okay, so these show DNase hypersensitivity valley, and this is a kind of a peak of DNase hypersensitivity, which depends on the structure of the factor. These are the peaks from HepG2 cell line, and he finds FOXA1 and HNF and HNF4 alpha as cofactors. And in the HUVC cell lines, ALK1 and AP1 and FEV are predicted to be cofactors, and GATA in the K562 peak. So in all the cases, these sequence features show enhanced DNase hypersensitivity and conservation. So in conclusion, I've shown that our gapped Kmer SVM model can predict enhancers from DNA sequence with high accuracy. And in addition, as opposed to our Kmer-based model, it can predict the fine-scale structures within the enhancer. We typically find 4 or 5 transcription factors bound per enhancer, and that these high-scoring peaks identify tissue-specific cofactors and show DNase hypersensitivity that's consistent with footprinting data. This cell-specific TF binding is the mechanism for the prediction of the enhancers. That can be predicted from the primary DNA sequence itself without any prior knowledge of when the cofactors should be active or what their binding specificity is. And that local sequence context controls this tissue-specific binding and the enhancer activity through cooperative interactions. And where it exists, the cofactor binding data supports this hypothesis. And finally, we showed that this generates novel mechanisms for direct validation, and our initial experiments show that luciferase expression changes are consistent with our model. So thank you.